everyone. Fred's pulling it up for me.
Awesome. And as a reminder to everybody, I'll be keeping track of any questions. So if you've got questions, please put them in the chat and we will address them when it makes sense.
Okay, let's get started. Thank you, Lee. I'm so pleased that you have joined us this evening for our 4 2019 grantees who are more or less finished with their grant year, although some setbacks because of COVID, and are here to tell you about the amazing things that they've accomplished, maybe lessons learned, and then to inspire you. When I ask grantees where they heard about our grant program, the thing I hear most often is that they heard it from our membership. So talk it up, let folks know we're beginning our grant year. We're advertising right now. In fact, this meeting is part of that marketing of the grant program. And the information on the website has recently been revised, the grant guidelines and the grant application. If there are any of you out there in the audience from another chapter, perhaps interested in starting a grant program, I would love to talk to you. Go on our website, look at the guidelines, look at the application, and let me know if you have questions. There's an email address in there for you. So I want to start with just a word before I introduce our 2019 grantees. I want to say a word about the grant program. In 2014, our chapter was growing leaps and bounds. We began to have to pull in more and more chairs for our programs, which are wonderful. But the concern the board had was that we didn't have membership participating as we had hoped. Our grant sales, our plant sales were also growing and booming, and we had funds to spare. And so it was suggested that a grant program might be a great way to use those funds and to encourage membership participation. And the volunteer appeal that you heard Lee announce at EcoCentro is for a grant that is currently in progress. So reach out to EcoCentro and volunteer if you have the time. So in 2015, a small committee of myself, Pam Peck, and Pam Spurgeon spent hours and hours looking at grant programs online and creating the grant program for this chapter. And our first grant year was 2015. In 2015, we had 2 grants. They were both 2 community gardens. In 2016, 4 grants, 2 school gardens and 2 community gardens. 2017, only 1 grant, but oh my, it was a doozy. It was George Ozuna's grant out at Medina River Natural Area. 2018, 5 grants were awarded, 2 school grants, 1 community garden, 1 university research project, and George came back for another project at Medina River Natural Area. In 2019, the year we're celebrating tonight, 4 grants, 2 university projects, 1 a research project and the other an outdoor classroom, and 2 community gardens. Many of you may not know that in 2020, we had to skip the grant program because of COVID. 2021, we had a small pilot program with San Antonio ISD. We called it Seed Money for Seeds, small grants for teachers to buy seeds for their native plant gardens, educational gardens. And then we had 1 traditional grant, and that was the one at EcoCentro. So let's get started. I will be actually presenting on the first of 4 grants tonight. Stan Drezick is the grantee. Stan is a longtime member, a very active member of our chapter. He's had some family issues and was not able to participate tonight, but we wish him and his family well. Stan took a brilliant approach by having a big project that he wanted to do, but he wisely has spread it out over 2, and now I understand he's going to apply for a grant this year in 2022. The site of his grant is the Inner City Development, which is a nonprofit on the near west side of San Antonio. They provide emergency resources, job training, food pantry, after-school programs, etc. They are on the corner of Chihuahua Street on the west side, and Stan wanted to replace their yard on those 2 streets with native plants, so that as people walked by Inner City Development, they would see native plants. And so in his first year, he planted the natives, put the beds in, and then he came back the second year. And in the second year, what he hoped to do is continue planting natives and then create labels for the plants. That education piece is so important. And create a bilingual brochure to get out to the neighborhood and the inner city development partners. Education, education, education. He had help from the Native Plant Society volunteers. He wanted to be sure that I mentioned these. Joyce Knappich came out for that first year, and that was actually 2018, and gave him ideas about writing the grant and plant selection. And then Joan Miller came out for this current year, 2019 year, and again provided help on plant choice. The volunteers who helped him plant were Inner City asked him how they could volunteer and assist, and Stan Dresik wisely said, "You can dig the holes." And so that's how he got the holes dug. And Jack Elder, one of our members, and his grandson, Rowan, came out and planted. You will see these folks in the little slide show that we'll show. And Norma Jean Moore came and spent hours and hours volunteering planting. In addition to the planting, he created signage in metal frames with metal stakes, and heavy laminated signs, putting the name of the plant and the culture. The culture information is so important for people who don't know plants well. They need to understand, does it need sun? Does it need shade? And then the brochure, the bilingual brochure, has been printed. They plan to hand deliver that door to door to 150 homes in the neighborhood, and of course to the inner city development partners. When we visited, I asked Stan, had he seen natives come up in the yards in the neighborhood? And he was so frustrated. He said, no, none. He said, I was naive to think that if we put natives in that yard around inner city development, that the neighbors would naturally pick it up. But in fact, there are very few resources for native plants in that part of town. You folks know perhaps Phoenix is on the south side, but that's about it. So Stan's got some ideas. He wants to perhaps give away plants or sell them at very low cost, do some tours of the gardens that he's created at Inner City. And that will be an effort to hopefully bring people a little closer to begin to experience the plants and the pollinators, the bees and the butterflies that are zooming around, and then informally educate on the benefit of natives. So if you have questions about Stan's project, please put them in the chat, as is the case with any of these presentations coming up. Okay, I'm going to shift then and introduce our next presenter.
You want me to show the video?
Oh, I'm sorry, I forgot about the video. Yes, please show the slideshow.
Okay, I will do.
This is Inner City Development Gardens. Thank you, Fred. And I know Stan would want me to say thank you to all the volunteers that participated in this project, both from our chapter and from inner city development. Okay, next I'd like to introduce our second native plant project grant that was in 2019. This is Dr. Kelly Lyons. She's a professor in the Department of Biology at Trinity University. She's here with her student, Mike Leonard. This is a research project, plant-associated fungi in the Edwards Plateau.
Thank you, Sherry. Let me share my screen. Okay. So I'm Michael Leonard. I'm a senior at Trinity University. I started working with Dr. Lyons in 2019, the same year that we got this grant. And like Sherry said, we've been working to characterize grass-associated fungi in mostly the Edwards Plateau, the hill country. For those of you not familiar with these fungi, they are a very diverse category of organisms. By definition, they spend part of their life cycle living inside plants without doing visible harm. And that includes a wide range of symbiosis, ranging from mutualism to mild parasitism, that I'll get into in more detail briefly. This is one of the types of the fungi that we've been working with, arbuscular mycorrhizal fungi, or we just call it AMF for short. They're one of the oldest types of these fungi. They evolved around the same time that plants and started to colonize land. They're very widespread. They're obligatory mutualists, which essentially means that their life cycle depends on having a mutualist relationship with plants. And what that looks like is aid in nutrient uptake. These fungi are really good at getting nutrients, especially phosphorus, out of the soil. So they do that, and then they basically trade it with plants for the carbohydrates that plants can make through photosynthesis. The other main type of fungi that we work with is called dark septate endophytes, or DSE. This is a highly understudied group of fungi. They're not well classified taxonomically, but they're very widespread. They're found in almost every terrestrial habitat. And unlike AMF, they have a broader range of symbiosis. Some of them help with nutrient uptake like AMF. Some of them are parasites, and some of them are saprotrophic, which means that they're more or less neutral, and they're just waiting for the plant to die so they can start decomposing it. So the ecology of these organisms are not well understood, but it is widely accepted that they are very important in the ecosystems where they are present. Here in Texas and in the grasslands, they're especially understudied, but there is evidence that they can help with things like drought tolerance, nutrient uptake with both AMF and DSE, and adaptation to climate change, which ties into drought tolerance. As you can imagine, with ecosystems getting more arid, that will become more important. And they're also suspected to affect competition between invasive and native species. This is all things that we consider to be of ecological significance, but they're not well studied. And our main goal has been to first characterize the community of endophytes, fungi, in these ecosystems so that we can get a better idea of what is present and then start getting into these topics. So the first step to that is culturing and isolating. So we go out into the field, we collect samples, we target grass species, mostly native and a few invasive. And then we take them back to our lab. We cut up the roots, put them on a petri dish, and then the fungi will actually start growing out of the roots onto the dish. And then we can isolate them, put them on their own dish and let them grow some more. And then we'll take samples of that and send it to some colleagues at the University of Tennessee who will then do some DNA work so that we can start classifying them into specific species, or OTUs is what we call them, occupational taxonomic units, because again, these are very understudied organisms and we do not really have a good species model for them. Unfortunately, because of the pandemic, we haven't been able to get the results back from this part. And we believe that we'll be getting it back soon now that things are starting to look better. And that's really exciting for me since this is a project I've been working on since I joined this lab. The other part of this study has been microscopy, which is how we estimate the abundance of fungi inside the plants. So we'll take roots, we'll clear and stain them, and we'll start using methods to estimate the abundance. The main way of doing that has been what we call percent colonization, where you basically look at how much of the root has fungi present in it. But we've developed a modified method that instead gets the density of fungi in the root. Like I said, the pandemic has really affected our progress in this, but I do have one graph I'd like to show you guys. This is not conclusive because we haven't really been able to dig into the data too much. But what we have here are, I think that's 5 species of grass. We got on the left KR bluestem, which is a pretty notorious invasive species, followed by little bluestem, purple threon, silver bluestem, and Texas wintergrass, which are all native. The light blue shows the density of AMF fungi, and the dark blue represents DSE. So what we can see here is that in most of these species, AMF and DSE are more or less equally present, except for in, I believe, it's hard to see the figure because of this bar. Hold on. Let's move that. Except for silver bluestem and Texas wintergrass, both of which have more DSE than AMF. As I mentioned earlier, DSE are extremely understudied, and this suggests that despite that, they may be of equal ecological significance as AMF. It also appears that little bluestem may have more fungi in them than the invasive KR bluestem, but we do not have high enough of a sample size to really confirm this, especially since, as you can see by these error bars, there's a lot of variation within these plants. So yeah, I've been working on this since a freshman, and now I'm a senior. So this project has been really significant to me in my time in college, and it has been really exciting. And I'm really thrilled to be able to start getting data back on it and starting to kind of see what the significance of these organisms are. So thank you to the Native Plant Society for your support. And now would be a good time for questions, or we can do it at the end too, if that'd be more appropriate.
There is one question that came in. It's probably best to just answer it now, I think.
Sure.
Do you think so, Sherry?
Yes, that would be great.
Yes, and the question is, in what ways do the fungi affect competition between native and invasive species?
That's a great question. There are, and it's also one that we are not certain on, but there are a number of hypotheses out there. One is that some of these invasive species need those fungi to be competitive. So it might not actually be the plants that are competing with each other, but the fungi in the soil. And the fungi that associates with the invasive species is able to outcompete the native fungi, and that allows the invasive plant to take over. There's also the opposite, where native plants will have parasitic fungi that have evolved specifically to target them, that have not evolved to target invasive species. So that means that the environment is actually more hospitable to invasives than natives. Those are the two main theories. There's also a lot of environmental factors that come into play. I mentioned drought tolerance earlier. With climate change, as environments adapt, if these fungi, specifically the ones that can give advantages that are important for dealing with climate change. I'm sorry, sorry. Yeah, so that's one other thing. And yeah, there's a lot of stuff like this, and unfortunately it just gets more and more complicated and more detailed. And a lot of these are really theoretical.
Thank you very much. That's the only question we had.
All right, thank you.
Okay, that was wonderful. Thank you so much for presenting that, Mike.
Thanks.
Really, really interesting. Appreciate it. We're getting questions in the chat about things you're seeing on the screen. We'll hold most of the questions until the end, unless it's a burning question, and then Lee will probably bring that forward. If you want any more information on the folks who are presenting tonight, you can email me at the grant email. That's in the chat as well. It's NPSOT, S-A, grants, plural, @gmail.com. Okay, let's go on to the next grantee from 2019. That is Dr. Rodolfo Valdez Barrias. He is Associate Professor of Biology at A&M University, San Antonio. He created teaching gardens to teach the native pollinator and host plant ecology to South San Antonio Independent School District high school students and his own undergraduate, A&M undergraduates, on the ecology of these plants. Okay, take it away.
Thank you very much. Let me go ahead and start sharing my screen. Can you all see my screen now?
Yes.
Okay, wonderful. Well, thank you very much for inviting me to share this. Even though it took a while because of COVID, I'm glad that I was able to present this to the Native Plant Society family. So as you see, the idea was to use native gardens as a way to teach concepts, whether it's ecology. Right now, we started as a pilot study using it for undergrads in their general biology course. And then we're planning on expanding these concepts on other projects that we're currently doing right now at our university. So the main objective or goal of my study was to establish a raised bed garden system that had pollinator-friendly plants, and to educate students, whether it's high school students, because we had a 4-H program that was about to begin at our university, and then, of course, our undergrads at A&M San Antonio. The second objective was to have an interactive educational program where they can actually see the plants. Many times we teach about them in the classroom, and this way they can actually sit outside and learn directly. The third one was to give our Native Plant Society volunteers a chance to participate in the process by giving us ideas and help. And they actually were very helpful for this. And also to integrate these gardens with a couple of beehives that we have already started at the university. So to give you a little bit of background, in 2017, a group of students asked me if we could initiate some activities on campus. So I reached out to my chair and we were able to get some space cleared. As you can see on the left side, there is a 0.5 acre land that was basically cleared for this project. I initially asked for 10 acres and of course got 0.5, but that's okay. So we started very humbly with a little tent just to start propagating some material, but that wasn't good enough. It wasn't going to work well. So we were able to get some funds to get a little bit of a bigger greenhouse. And of course, with the help of HEB, we were able to also get somebody to put it together. And now we have a relatively modest greenhouse to get all these projects started. We also thought of initiating the gardens with vegetable gardens first. So we introduced the students to things they can eat first, because it appears that they're less knowledgeable of native plants than edible plants. So the first place where they interact with the plants is at the vegetable gardens. And we have a total of 10 raised beds. Recently, we got some elevated tables that are wheelchair accessible too. And then, of course, after that, we got the beehives. This is the project of Dr. Alan Daniel, who is a psychology professor that uses bees to study different behavioral components of human physiology, like addiction to sugar and things like that. Very interesting, cool study. So he uses those bees. So he thought of setting that beehive near our gardens. So then I went ahead, and in order to make that happen, we already had the vegetable gardens, we already had the greenhouse, and I thought we needed the native plants. So my program was able to put in some money for the beds that you see there. And then I reached out to the Native Plant Society for a grant to mainly help me with the purchasing of the plants and give me some ideas on what plants to get. So we went ahead and had the volunteers come over. That's one of my students' volunteers. One of the groups that has been very helpful has been the Native Plant Society group, especially Fred Loxham and Jerry Morrissey. They've been very, very helpful since then. And to this day, they're helping a lot. Also, our plant club, I started with a group of students, the Texas A&M Plant Club, and they've been very helpful as well. And now we have other organizations within the university that are helping me maintain these gardens. So as you can see, we went ahead and then put our benches so we can seat about 18 students at a time. And then we put all the material in the center so that we can do rotations and put different species, and then have the students basically sit in front and do observations directly on the plants. So for this material, I actually used the $500 from the grant, and was very surprised by being able to go to the Medina nursery, where Ernesto and Ismael gave me very good tips on what plants to get. So all the material that we have on those beds comes from the Medina nursery, thanks to Fred Loxham, who recommended that place. So here's the breakdown of how we use the money. We use some of it for mist irrigation kits so we can propagate materials inside our greenhouse. We use some plant growing trays and some containers to grow those plants indoors so that we could keep, growing some of them in our own nursery in the greenhouse. And we purchased about 30 native plants from the Medina nursery. So to give you an example how we use this, here's an example of the laboratory that we do with our students. So this is for general biology 2. So in general biology, it's a little bit more organismal and ecological than general biology 1, at least at our university. This activity was, of course, modified from another activity, but we've been doing it for a while, and we thought it was very appropriate for this kind of study. So we basically have them try to understand the relationship between pollinators and plants. We have them try to identify the plants. When they are shown the plants in front of them, we give them some resources on which plants are present. We don't label them, so they're supposed to figure out what they are. And then once they figure out what the plant is, they start observing to see what pollinators the plants are actually attracting. So this is sort of how we work it out. I'm not going to go in great detail, but we split them in groups of 2. So you have one person that is the observer. The observer is allowed to go up and go around their bed. So we have 2 students per bed. They're each going to do a counting and a characterization of each plant and try to not get too close so that they don't scare away the pollinators. They're supposed to watch it for a certain period of time. And then, of course, observe visitation. Because most of these students haven't had any, advanced entomology, we just asked them to classify them as Hymenoptera, Diptera, Coleoptera, Lepidoptera, just basically by order. We give them drawings so they can actually recognize what those insects look like. And here's an example of the spreadsheet they use. So here are the different taxa, the different orders that they're supposed to work with. And here are the species that we have currently on our raised beds. The Maximilian sunflower has grown pretty well. And some of them are not doing very well. So we've had to replace it with other species. And so most of these have worked well. And the Zizotes, for instance, we were able to get it established only for 1 year, thanks to some collaborations with the Medina Natural Area where they showed us how to harvest in some empty fields near the south side. And we were able to plant them and grow some of those milkweeds there. So as you can see, this is the activity. The students learn the scientific name of those native species. You might notice that some of them are more typical of the hill country area. I sort of did that on purpose so we can start the labs a little bit earlier in the semester, because I've noticed that if you have material that is grown in the hill country area and you bring it to the south side of San Antonio, it's about 600 feet difference. It does affect the blooming. So I was able to get plants to bloom a little bit earlier by doing that. So what was so nice is that on our first time with the students, visitation was pretty good. But these are some of the selected photos. We've had the swallowtails come over, and of course, some of the queen butterflies. Actually, the day we were planting them, all of a sudden, I don't know how they knew we were putting plants. And every time we water, I don't know what is it about the watering process, but every time we water them, the butterflies seem to notice that something is happening and they'll come and visit. And of course, we have the bees from the beehives that are significantly helping. So here's kind of how it looks right now. And right now below you have the students from the Native Plant Society. I'm sorry, these are the students from the plant club at my university. And they're here looking at one of the Zizotes milkweed that we were harvesting. What we tried is to not only have them work over here in the gardens, but to start learning, do activities in the field, help me collect some of the seed in the field. And of course, we're in the process of resurrecting all of this because during COVID, everything got shut down. So the garden, we're gonna have to do some major purchases this spring because we were not able to start everything from seed. So yeah, in the future, if you wanna think of a place where plants are needed, definitely here's a garden where your help will be very well appreciated. And that's it. That's everything I have for today. Thank you.
And we do have a few questions. Do you want to get those done right now, Sherry?
Sure, that's great.
Okay, Rodolfo, you ready?
Yes.
The first question is, what was the land use before the garden plot?
That was an area with mesquite. It was sort of like a secondary growth area behind some portables that we had at the university. And so I actually didn't pick the spot. The university said, I said I want 0.5 acres, and they went and bulldozed some of those. So that was supposed to be basically mesquite and research that got cut down to create that space over there. But unfortunately, just to tell you, we're losing that space in about a year because a huge building is going to go over it. So we're in the process of relocating all of this to a new home, maybe near the student dormitories. I think it's going to be closer to them and more appropriate. So, yep. So that will get moved probably in about a year from now.
Might need some volunteers for that.
Oh, yes, of course.
Okay, next question is, someone's curious about how the Plant Club student organization started, was it from a previous project or from student interest, or how did it get started?
Yes, first it was called the Plant ID Club. It was students that would go to or actually we would go to the Texas Society of Ecological Restoration to compete with them. And we had some awesome students that did really well, like Sarah Galvan, that many of you probably know. And so the problem was that we had large membership, but only a couple of students would compete. So the students said, can you broaden it? And instead of making it just Plant ID, can we make it a vegetable garden or a plant club? And so the group of students you see there below it's one of the group that kind of initiated it. So I always have an average about 4 or 5 students. I get them more or less during their sophomore year. And I get to keep them for about a couple of years until they have to, switch to another cohort. But they have been the heart of this. Everything you see out there has been done with the work of students. Probably Saturday mornings. I don't know how many Saturday mornings I've spent in the last 3 years with them, but it's yeah. So that's my answer. Sorry.
Awesome. Okay, and then there's an elementary teacher, I believe, who's wondering if you would be willing to share photos of your project so that they can show their elementary garden club. And they think that it'll be good for them to see that college students are also doing the same thing. And she included an email for you. So the main question was whether you'd be willing to share photos with her of your project.
Anything, yes. I will be more than happy to participate and collaborate with this. So yes.
So that was Catherine Jimenez. So we'll make sure.
And I can send that email to Rodolfo.
Wonderful. Thank you.
That's it for the questions.
Okay. Thank you very much.
So you'll need to stop sharing, Rodolfo. Excellent. Thank you.
Rodolfo, thank you so much.
You're very welcome. Thank you.
You folks are getting lots of love in the chat, so be sure and look at that. Folks are really enjoying these presentations. For our final grantee from 2019, I want to introduce Lilyn Koch. That's K-O-C-H, Koch. She's from the Bethany Congregational Church, and her grant was for a community pollinator garden. This project has a strong educational component as well. Okay, Lilyn, take it away.
Okay, and I'm relying on Fred to take care of the PowerPoint for me.
Okay, I'm going to start it up right now.
Okay. And while he's doing that, I just want to say how exciting it is to listen to these other projects. It's very inspiring, and I'm very pleased that you invited all of us to come tonight to share our progress, because I think we each take a lot of pride in what we're doing, and it's nice to feel appreciated. So Fred, I want to ask you, are you certain that this is the one I sent you today?
No, it's the previous one, so I hope it's the same.
It is not the same.
Okay, I can get that one. If you want to talk, I can get the latest one then.
Okay, I will go ahead and give a little background in history. The Bethany Community Garden was started in 2014 by the youth group of Bethany Congregational Church. It had not had problems attracting volunteers of all ages. And we are part of the Green Spaces Alliance network of community gardens. And back in 2019, we applied for a grant from the Native Plant Society, because I think, as most vegetable gardeners know who are trying to grow things organically, pollinators are a very important part of that process. And people who grow food that way are also usually very ecologically minded. So we broke ground in late July of 2019, and we had a crew of both church volunteers and Native Plant Society volunteers that helped us. And how you doing, Fred?
I'll be another second, another 10 seconds or so. We hope.
Okay. Okay, so when he gets that up, we'll show some pictures of the process we took. We had to sheet mulch the area. Our space is full of Bermuda grass. So we collected a lot of cardboard and laid that on the grass, watered it really well, and then covered it with mulch. And yes, this looks right. Thank you, Fred. So you can go ahead and forward to the next slide. So here, this was actually an interesting, cool day in July. It was surprisingly cool that day. So everybody was in a really good mood. So we prepped the cardboard, taking the tape off it, and next slide, please. And we had all that cardboard we laid out. We had marked the space with, marking spray, and we watered the cardboard really well. Next slide. And then we had next slide, please, Fred. Thank you. We had a trailer of mulch delivered, and we all laid that, and everything went surprisingly well and efficiently. And you can go to the next slide. It's a group picture of our work crew that day. It's about half church volunteers and half Native Plant Society, and some of you may recognize some of the native plant people. So very grateful to that crew that came out and made it so easy to get that done. Next slide. We had a volunteer named Rob Flynn who very knowledgeable with native plants. He's previously worked for one of the river authorities and also AgriLife. So he did a lot of the propagation himself at his home, and these are some pictures of that. Next slide. And then this is planting day, where he showed up with a SUV full of his plants, and I know he purchased some plants as well. So this is the very early stages. Next slide. And installing the sign, and you see all the plants here are fairly small. And then you can forward to the next one, please, Fred. So it's been 2 years now, and the plants have really come into wonderful maturity and profuse blooms. So Rob got some very great pictures of some of the visitors, some of the critters that have visited the garden. Next slide. And this hawk on the bottom left was a juvenile red-shouldered hawk. He tells me he named it Hope, a fitting name for COVID times. Next slide. And more critters. And I want to talk a little bit also about the human visitors that we've seen come through. We are situated right next to Delview Community Park, and we get a lot of foot traffic through our vegetable garden and this space where the pollinator garden is. And one day, a mother and child came by, and she told us that every time they go to the park, her son insists on coming by to see the butterflies. We also have a Girl Scout troop that meets there, and I've seen them just become mesmerized, just standing for hours watching all the activity in the pollinator bed. Next slide. And just some more pictures, more, caterpillars, and just a ton of blooms. Next slide. This is what it looked like this past fall, about October. They're right in the center. There are very large stands of Maximilian sunflower, which stayed modest much of the time, but boy, this past year, it just really took off. And we have decided to remove that. It's just a little bit too overwhelming for this space. This is sort of the shady spot side on this side that we're looking at. A lot of shade tolerant plants, Turk's cap, autumn sage, cedar sage, standing cypress. And on the back side is where it was super colorful with mist flower and I forget a daisy. And so we're thinking we need to have a little less height there so you can see the entire space in entirety from every direction. We're still going to use the Maximilian sunflower around our compost area, sort of to hide the work area of the vegetable garden, because we did let the sunflowers go to seed and they were full of finches. I mean, that was exciting in itself to see all the finches being so busy gathering their seeds. Next slide. Oops. This is how it looked just this past weekend. So we had a volunteer starting to remove the dead material, and you can see already there is some green material coming back up. So we're looking, this will be its 3rd year. All the perennials did survive the freeze. So a lot of these plants, I'm thinking, are pretty well established at this point. And next slide. I want to talk about our education activities. We had planned to have 2 seminars a year, and those would have been in conjunction with Green Spaces Alliance. We were able to do 1 virtual seminar. Rob led that session, and he talked in length about the native plants, since that is, you know, his true passion. And they did the advertising for us, and they managed the technology for us. They also funded a sign for us, and Rob found some educational materials to put on the sign. And then all of our plants are labeled with the, you know, what everything is. Next slide. Oh, that came out kind of funny looking, but this is I think most everything that we planted. We got quite a bit of diversity, I feel like, in that relatively small space. Next slide. And then some of the lessons we learned that might help other people wanting to start beds from scratch. We did not quite have as much cardboard as we should have. Some of it was just in a single layer. So I would say save about twice as much as what you think you need, because you can always put down 3 and 4 layers and that would be fine. I already talked about the Maximilian sunflowers. They're a great plant, but find the right place for them. We do need to do some refurbishing on our plant signage. Some of them have kind of, they're, you know, sitting at an angle. Maybe they're hidden by the plants as they grew larger. So we're going to be working on that now that things are kind of cut back a little. And then we had some problems with leadership. I had to leave and go out of state for about half a year. Rob was able to carry the ball, but then he had some family issues just within the last couple of months. And we do have 2 new stewards at the garden. We have a husband and wife team who are also very knowledgeable with native plants. So we've been fortunate, but it just stresses the importance of making sure that you keep everybody educated so that when you do have absences, someone can pick it up and at least help you through a transition. So we managed to get through it, but it's just, I think when you're doing something run by volunteers, it's very important to make sure everybody's getting some development that they can help provide leadership if it's needed. And last slide, I think, Fred? Yes, thank you. So we're very proud of our garden and the amount of attention that it gets. Very happy to talk to the people who pass through and see the excitement they see with the garden. And we look forward to being able to educate the community even more with some formal lessons as things open up a little more. So thank you so very much for your support and your gift that made it possible for us to put that together.
That was awesome. Thank you. We do have some questions.
Sure.
So go ahead and start with the first one. And the person was wondering about your process. So you put the cardboard down first, then you wet it down, and then you put mulch on top of that, and then you planted. Was there a certain amount of time you had to wait before you planted, or did you plant right away?
Yes. Yeah, no, that's a very good point. We planted in July. We did the sheet mulching in July. We might have kept it watered a little bit during, August and September, but pretty much it was that way through the winter, and it was like midwinter, early, I'd say December through February when those plants started going in. I think Rob did mention that some spots were a little hot. I should have put that in lessons learned, but there were some places where maybe it wasn't as fully decomposed as it would have been. So I think maybe allowing maybe 8 to 9 months to let that break down before you plant.
Thank you very much. And then there's another comment here or question about you showed some photos of what the plants looked like this past weekend and mentioned that it was going to be cut back. And they're pointing out that there's some advantages to not cutting back everything that's dried up, and wondering if you had considered the benefits of leaving some of that material.
No, I'm really a beginner and a newbie, so I would appreciate the thoughts about that. Tell me more.
That was a great comment. I think that was from Patricia. But if you leave some stems, for example, 9 to 14 inches high, instead of cutting all the way to the ground, if you leave some stems, then this next growing season, some of the solitary native bees might come and use those stems for their larvae. And then they would come out a year from now if you just start looking at leaving some of those stems, and or maybe some of the leaf material down on the ground for all those little critters and organisms that are overwintering still. Maybe there's some other larvae or eggs or some things just to leave material. Or maybe the best way to think about it is don't mess with it more than you need to. You might need to remove some, but if you can leave a little bit, especially some stems a little bit higher, and some of that leaf matter on the ground, it can be a huge benefit for wildlife.
Okay, I will definitely. We've just barely started. Maybe only have about a fourth of it worked on. So for the remainder, I will tell them to leave some. You said about 9 to 10, 9 to 12
well, so
inches.
They say, somewhere, anything is better than cutting it all the way to the ground. So, and aster plants in the aster family tend to be used a lot more than others. They have a softer center, and then the outer tube part stays longer. So they just tend to use those. So anywhere from like 9 to 12, or if you can leave even taller, but the plant will grow and hide those stems, and so it won't be unattractive for very long, but it can be really beneficial for those solitary bees. And I'll ask Patricia too, since she asked that question, if I missed anything that she was thinking of, because there's a lot, that can be beneficial to kind of just minimal. You save your own resources too, and then you don't have to worry about what you're going to do with all that material, right? If you don't have to mess with it. So
thanks for asking, Lee. I think that I learned that from Joan Miller telling me, pointing out in some of the native landscape certification classes, and even in her own yard garden, just like, look how the spiders are using this to make their webs. Look how birds are gathering some of the dead material for nesting material. Sometimes, the grasses or something that bunch grasses where they might actually be nesting in the middle of that bunch. So we're kind of unaware, and cleanup is part of a human thing to do, and nature doesn't clean up after itself.
Well,
yes,
I appreciate that. Those are very good points, and that's the learning opportunity for us, and that'll be an opportunity for us to share that with other people too, with some of our visitors. Thank
It
you.
is. So there's, if you want to look it up, if you just search for Leave the Leaves, it's a Xerxes Society
Okay.
campaign. And another insect that benefits a lot from that is the lightning bug. So
Oh, nice.
lots of different insects could benefit from it. And
Right.
that's it for the questions. There's a lot of comments about everybody and everybody really enjoying these presentations. Sherry, did you want to close it out for us, or?
Yes. Let me just say thank you to all of the presenters. Wow, the projects are so different and yet all so interesting. And when I think about the number of people that have been served by these projects, whether they were students who participated in bringing it together or the passers-by, like Lillian talked about, who popped in and took a look at the critters in the garden. I'm just amazed and really proud and pleased with our chapters program. Thank you all for participating. And if you know anyone who's interested in a native plant project, please steer them to our website and the grant application, guidelines and application.
And Sherry, thank you so much for chairing this committee and being such a great leader for all of these amazing projects, and for leading this tonight. Thank you.
Appreciate it.
And everybody have a good night. Thanks for joining us.
Thank you, Sherry.
Thanks, Fred.